Good afternoon, everybody, and thank you so much for coming to the public forum. It's a great pleasure to welcome so many of you here. As you probably know, we're capped at a certain number because of fire regulations, but it's still really great to see. We were basically at capacity, and we're also live streaming out, and I think there's probably 100 plus people online for the live stream, which is just terrific. For those of you who don't know me, I'm Simon Lewis, and obviously I'm very proud to be here at Macquarie University and have a research program that's very active, which we're going to hear a lot more about today. It is one of those sessions where you're going to it'd be useful to have your mobile phone to hand, because we're going to have some QR codes for you to scan, so don't be afraid to go scanning QR codes. Before I go much further, I'd just like to pay my respects to traditional owners of the land, past, present, and emerging, and acknowledge that on behalf of the university as well. We've got a packed program, but hopefully enough time to enjoy some refreshments and some good conversations and networking. The plan is that I'm not going to ramble for too long, I hope. But in our first session, the idea of today is to really show our bright young things. The talent that we have, young researchers, young clinical fellows, who are hopefully doing the sort of research that we need to change the field. So we'll have 4 presentations. I'll introduce the speakers as they come through. And then after each one of them is presented, we'll actually roll into the next presentation and take questions at the end. And we'll get the guys up and we'll do a bit of a panel. And we'll have a couple of roving mics for questions. And hopefully we'll be able to respond to your questions with answers. And we'll then have a break, which hopefully will give you the best part of an hour. And you've already seen that we've got some bathrooms, and we'll have some refreshments coming out for mid-afternoon tea. And also, just to make you aware, if the fire alarm does go off, then the idea is to move out of the building, out to the front, and assemble in the car park. I'm not sure if that's the real plan, but that's what we're going to do. Hopefully it won't come to that. And then after the break, we're going to have 3 more presentations, another Q&A, and hopefully get us to the next point where we can tell you about what's going on next. I want to put this slide up to remind me to talk about the research and the opportunities. So probably the biggest trial we're involved at the moment is the Australian Parkinson's mission. So this was $30 million out of the federal government and running across Australia. So the first trial has already finished recruitment. We'll have the results of that study probably in Q1 next year. The second trial, though, is going to end recruitment at the end of this year. So if you're someone who hasn't been in this trial, we're looking for patients who are diagnosed with Parkinson's, stable on their Parkinson's medications, who are willing to come in and participate in the trial. If you scan the QR code, it'll also basically allow you to say, this is what I'm interested in. It'll also say, I'm actually not interested in the trial. I'd like to do research, but not this. I actually don't have Parkinson's, I have something else. And you can tell us through this QR code. The other part that I wanted to flag, and you probably found these when you came in or on your chair, is that we're also very interested in hearing about your views about research and especially trials. This is what they call in the trade patient advocacy. So, for example, one of the things in my role with the Australian Parkinson's Mission is that I'm trying to attract companies to come to Australia with their opportunities, trials to see if we can slow disease, trials that maybe improve symptoms. And they come with all sorts of ideas about what's going to be acceptable to you, because they think they're helping you, which is great. But it may well be that what they think is okay for you, you might think is a bit too much. You know, they may want you to, spend 2 hours doing something or have a brain scan, and we just really want to gauge your level of comfort about what you would do. So you'll see there's a bunch of questionnaires you can fill in. Hopefully it's a tick the box thing, mainly just to make life easy for you. And when you've completed your questionnaire, they're anonymous. Just drop them on the front desk where you registered to come in. But we appreciate the feedback. That would be very useful to us. It would be remiss of me not to take the opportunity to plug an event. So I was humbled earlier this year when one of my patients' wives approached me and said, look, I've done some charity events before, and I'm very keen to do a gala dinner event for your research to raise money. And so that working with Macquarie University, we're very pleased that in February next year we'll have a gala dinner at the Dalton House, which is sort of next to the Star Casino, that part of town. one of the really cool parts about that evening is that we're going to have some entertainment for everybody. So it just so happens that the son of the patient is a professional singer, and he's partnered up with a young lady, and they travel around the world basically with their tribute act to Andrea Bocelli and Sarah Brightman. So I'm hoping that the guys can adjust the volume down if it's going to come out blasting too loud, but just so that you know what the level of entertainment looks like, this is their show reel. So they will both be there singing live for our entertainment. As you can see, they look like they've done it before. And the most important part of the evening is going to be the silent auction, which is to keep me silent so that I don't sing. And we just let these guys do their thing, okay? So that's the QR code if you wanted to attend the gala dinner. We've also had some fantastic sponsorship from patients and the like already coming in. So tickets are selling quickly, which is fantastic news for us. So February the 21st in the Dalton House. So it'd be great to see as many people there as possible. The other event I just want to tell people about is happening next Friday, and this is what they call a flagship event for Macquarie University, really focusing on what Macquarie University is doing in the space of neurodegenerative disease. So there will be a panel discussion where I'm on the panel along with colleagues who are doing work in motor neuron disease and in Alzheimer's dementia, and you'll recognize, no doubt, Ita Buttrose, who's going to be the plenary speaker on the day. Now, Mrs. Buttrose, many of you may know, came out publicly, I think last week, about the fact that she's been diagnosed with a Parkinsonian condition. She has a rare Parkinsonian condition, which I was tangentially involved in as a second opinion. For me, it makes life a lot easier because people now know what she's got, and that's fine. But she's going to be a wonderful speaker, and as you know, she's a patient advocate for Dementia Australia, and now she's really talking from a position of lived experience. So you'll see another QR code there. It's now been open to the public, so if you wanted to come along and enjoy that event, we'll also have some of the students at that event showing some of the research toys that they'll talk about today. You'll get a chance to play and feel with them, and if you've never had your sense of smell tested, I think they're going to be doing that as well. And then finally, in terms of the QR codes, it's always good to prepare. So one of the things I'm really trying to do with our new platform is to say to ourselves, okay, we want to do 2 of these public forums a year. We've done that now for the last couple of years. You'll have seen the video playing before with Roger Barker, who was here in April. Next April, that date being as close as we can in a working week to World Parkinson's Day, which of course is April the 11th, commemorating James Parkinson's birthday. But we've already lined up 2 excellent speakers. So Natalie Allen is a physiotherapist. She's done a lot of research with my team already, looking at the benefits of exercise and also how exercise can modulate pain in Parkinson's disease. So she's a wonderful speaker. And then Florin Gandor, who's doing his best there to look like a Bond villain, is our other speaker, and he's going to speak about, and he is German, so it's going to come across a little bit that way, but he's a very, very funny guy. So Florin is going to speak about the smart therapies in Parkinson's disease. So after the pills are no good, what are my options? You know, things like surgery of the brain, the pump therapies, new things like the focused ultrasound, new things that we're not even at yet, the stem cell work. So the aim is to really have a diverse session when we meet again in April. So hopefully you've had a chance to scan the QR codes. We'll have them rolling again through the breaks. And I wanted to just also welcome all the people who are watching. Presumably I'm being recorded. Hi. Do I look okay? Good. Excellent. And are at home watching this. So hopefully we're not getting any complaints, Louisa, from the feed. Good. Excellent. People are very good at that. And in terms of me advancing this for our first speaker, do you want me to just hit this? And that says end of show. So how do I bring up our first speaker's slides? Or are you going to do that from the back? You're going to do that. Okay, why don't you set up your slides and I'll introduce you. So our first speaker is Ajay. So Ajay has been with the team now probably a year or so. Almost 2 years. Almost. Close to 2 years. Time flies when you're having fun. Supported by the Rotary Club of Belconnen for his PhD. And Ajay is going to talk us through what he's been looking at. And I guess what I'd say to you is that Ajay's talk is probably the most technical talk we have today. I had to watch it 3 times before I thought I understood. It was a bit like when Einstein traveled to America. And when Einstein arrived in America, the guy that they'd had escorting him said, "Oh, how did you go with the journey?" He said, "Well, Einstein explained the theory of relativity so many times, I really think he understands it now." So Ajay's work is really looking at our diseases in a different way, because I'm a clinician. I look at patients and I make a diagnosis based on what I see. Ajay's research is saying, I don't know if that's the right way to do this, because in actual fact, some of these patients, when you look at their brain, despite the fact their diseases are different, their brains look very similar, doc. So are you sure you're treating these patients in the right way? If you're going to do a disease-modifying therapy, can we actually get maybe the right patients in the same studies? So basically, Ajay is trying to open our eyes. So please welcome Ajay Konuri.
Thanks, Doc, for the introduction. So good afternoon, everyone. Thank you so much for coming for this year's public forum. It's awesome to see you all back here again. And as Doc introduced, I'm Ajay. I'm a second-year PhD student working with Professor Lewis and the lab here. And I would be talking about, I will give you a brief update on the current literature, and then I would talk about the issues we face as a Parkinson's disease community, and then I'll use some of my results to show how big data can help us understand Parkinson's disease. And everyone in this room would agree upon something, that it is extremely important to tackle Parkinson's because it is the leading cause of disability. It is the fastest growing neurological disorder. And most importantly, we have limited knowledge on underlying biological mechanisms. So we don't know how the disease progresses. However, there are some things we do know about Parkinson's disease because we have been researching a lot in the recent years. So I would be talking about the clinical presentations in a bit. And there are some certain things that we know about cell loss and pathology, about the neurodegeneration we see in these diseases. So we do know the specific brain regions that are affected where we see cell loss. And we do know the specific brain regions where we see the accumulation of alpha-synuclein. Additionally, we also see the neurotransmitter systems or the chemicals in the brain that are often implicated in these diseases. And most interestingly, we have identified a prodromal group or an early stage indicator or an at-risk population that go on and develop Parkinson's disease or dementia with Lewy bodies. So this is IRBD, and the talk after me, Lachlan would be talking about it in more detail, but I would be touching upon the clinical presentations in more detail during my talk. And these are some of the papers that our lab have published, and they have kind of summarized all of the blocks that I mentioned here in a much more detail. So moving on to things that we already know about Parkinson's disease. So first of all, we do know Parkinson's disease overlaps with other diseases. So these include Parkinson's disease dementia and dementia with Lewy bodies. So the distinction between Parkinson's disease dementia and dementia with Lewy bodies is done by an arbitrary 1-year rule. So if a patient has motor symptoms like slowness, rigidity, and they go on and develop dementia after a year, then they're diagnosed with Parkinson's disease dementia. If a particular patient has cognitive impairment or dementia, and then they develop for a year, and then they develop motor symptoms, then they're diagnosed with dementia with Lewy bodies. So collectively, all of these diseases are called as Lewy body diseases. So to add complexity to these existing conditions, we will now talk about clinical presentations. So I've already shown you that Parkinson's disease overlaps other conditions. And so when a clinician diagnoses them, they usually diagnose with either Parkinson's disease or a Parkinson's disease dementia or a dementia with Lewy body. But the problem is they're so overlapping with each other, it's extremely difficult for a clinician to diagnose them right. And you might be wondering what's the rate of misdiagnosis in these conditions are. You would be shocked, but it's around 30%. So 1 in 3 people are being misdiagnosed, which is extremely important, which is extremely why it is important to understand the clinical presentations. So I was at a talk last week, doing a talk for my to my funders, and I asked them, what's the first thing that comes to your mind when you hear Parkinson's disease or Lewy body diseases? And they said, motor symptoms, the movement problems, or the slowness, rigidity, and tremor. But they were shocked when I mentioned some of the non-motor symptoms that we usually see in these patients. So visual hallucinations, cognitive impairment, mood disorders like anxiety and depression are very common in these diseases. So this image, I kind of love this image because like it kind of summarizes about how heterogeneous or how vast the clinical presentations are. So usually people think of the top of the iceberg, but again, there are a lot of other conditions that or symptoms that patients usually present with. So you can you would know how complex this disease is, and so which is exactly why we still don't have any cure. So as a summary of this slide, and most of the people in this room would know that no two patients would present exactly the same clinically, which is exactly why we still don't know some of the things. So what do we don't know? So the first, as mentioned before, we have limited mechanistic insights into disease progression. We don't know the underlying biology that's happening in the brain that is causing the disease to progress. We still don't have any disease-modifying therapies. We don't have any cure yet. And we don't know what happens at early disease stages. So this is before the patient is diagnosed. I mentioned about the heterogeneity briefly, and then I would give you some case studies. So these are not our patients. These are, and the pictures are just from the internet. And so I'll give you a case study. So remember these patients, because we are going to help them by the end of this talk. And if in the audience here, if you relate to any of the symptoms, maybe remember that particular patient. So the first we have Oliver. So he was diagnosed with Parkinson's disease 5 years ago. He has severe cognitive impairment. He has visual hallucinations, and he has tremor. And then we have Greta, who was diagnosed with Parkinson's disease again 10 years ago, but she only presents with tremor. So Greta was diagnosed 10 years ago, but still only has tremor, whereas Oliver has other symptoms as well. And then we have Charles, who was diagnosed with Parkinson's disease 5 years ago, and he has mild cognitive impairment, RBD, and visual hallucinations. And then we have Oscar, who was diagnosed with dementia with Lewy bodies 3 years ago, and he has dementia and moderate Parkinsonism. So you can see how distinct the symptoms are, or how variable the symptoms are. So if we consider this room, the patient sitting next to you would not have the similar clinical presentation, which is exactly why we still don't know certain things about this disease, and we are trying to tackle them. So I mentioned about the clinical variability or the heterogeneity. And we have been banging our heads against the wall to solve this clinical heterogeneity. But recently, the recent literature suggests that we might have found a way to understand why these patients present so clinically different, which is basically subtyping the patients. So what these papers say is we have been looking at Lewy body diseases or Parkinson's disease as a single disease. But what these papers suggest, and then we are continuing some work after this, is basically saying Parkinson's disease might not be a single trajectory. It's not a single disease. There are some subtypes in it, or subgroups of patients, which present similar clinical presentations or similar disease progression trajectories. So I would explain more about the subtyping in detail. So imagine a pool of mixed color balls. So these are patients with Parkinson's disease. So if you select one particular ball from this, and you are trying to understand Parkinson's disease, I don't think anyone in this room would be able to understand or get a complete picture of the disease, because the ball next to it is so different. How would you get a complete understanding? So what we did was we basically subtyped it using an unsupervised machine learning technique. And then what we found was we clustered the colors in it. So you're seeing a lot of colors in the first image, but now it's kind of like clear. We got all the blues, the greens, and the yellows together. So these are called the subtypes or clusters, which have similar disease progression or similar clinical presentation. So this makes more easy for us as researchers to interpret what's happening in this particular subtype, because they are similar, unlike Parkinson's disease as a whole, which is so different. So we went on to do another step of analysis, which was staging. So if you see in the first subtyping image, the colors of the balls have different shades. So the shades indicate the severity. So the light shade indicates they have lower disease staging, and then the dark shade indicates they have higher disease staging. So we were able to subtype and stage patients. So for the first time, we had a subtype and a stage for all of our patients that were included in the study. So we knew what subtype that they would have. Even if they're at early stage, we could predict what symptoms that they would be developing. by the time they progress. So, which is interesting, and this image kind of like puts up the differences between subtypes in a much better way. So in the first image, if you see, so this image basically says that in subtype 1, brain region 1 progresses quicker, and then in subtype 2, brain region 3 progresses quicker, which is exactly why they have different clinical presentations. So that was kind of like a brief technical knowledge on the subtyping that we did. And thanks to Professor Simon Lewis here and Dr. Eli and all of our collaborators, we were able to collect 1,250 MRI scans from across the world. And then we estimated atrophy or cell loss in these 1,250 patients. And then we, as mentioned before, we applied a unsupervised machine learning technique, which we call the sustain, to kind of like subtype and stage patients. So what did we find? We found some interesting results. So I'll explain this image a bit. So these are the left side of the image is basically the clinical diagnosis a usual clinician would give. So when a clinician gives diagnoses a patient with Parkinson's disease or dementia with Lewy bodies, they might or might not represent the clinical symptoms. But what we were saying is, hey, I think the subtypes are a better indicator of the clinical heterogeneity that we see, and they exactly reflect the symptoms that the patient is presenting, which is exactly what we need when we're trying to develop treatments. Because we need to know what symptoms they present with, we need to know what stage they are in, and we need to know what subtype they are in order to develop personalized medicine. So if you remember the patients from before, they align with each of our subtypes. So if you remember Greta, she was diagnosed with Parkinson's disease 10 years before, but she only had tremor. She didn't have any cognitive impairment. So here, we were able to show that Greta is kind of like part of a slow progressing subtype, which is subtype B. So this kind of like summarizes how subtypes could be a better indicator of developing targeted medication and personalized medicine. So we did all of this. We were able to find subtypes and stages just of structural MRI. I know most of the patients would have MRI here. So structural MRI is basically taking a snapshot of your brain. So we're just taking a picture of your brain, but there are a lot more things that we could do. So for example, there's functional MRI. So functional MRI looks at your brain over time, how your brain connections are talking to each other. And then we have diffusion MRI. So diffusion MRI is basically looking at connections between different brain regions. And then we have PET scans, which looks at the energy landscape of your brain. And then, so these are just part of imaging. And then we have fluid biomarkers, genetics, and pathology. And we need all of these biomarkers to get a better understanding of the disease. And this is exactly what we mean by big data. And the big data is you patients constitute the big data. And we need your help to kind of like develop a bigger data set. We need more biomarkers to understand this disease in a more comprehensive way and develop personalized medicine. So to end that, I would first thank all of our patients who are part of our study. And thank you so much for contributing. And I would like to thank the supervisors and the lab members. And especially special thanks to Rotary for funding my PhD. And yeah, thanks so much.
So that's why we put him on first. Your brains are now warmed up. And he is available for questions, which is grand. Our next speaker is Lachlan Churchill. So Lockie's basically going to take us through some of the work he's done about how do we predict people who we think are going to get the disease so that we could treat them before they get the symptoms that we've just heard about. And then how do we perhaps use that in our clinical trials? Thank you, Chris. Our clinical trials to be able to say, OK, if you've got someone who doesn't have a disease, how do you measure the progression of your disease if you're in a disease-modifying trial? Because if they don't have tremor or slowness, you can't say, well, it didn't get worse or better. So what we want to do is find clever ways that we can actually look at those patients who are yet to be diagnosed. And I come from a sporting background, and my sporting background tells me retaliate first. So this is what we want to do with our world. So please welcome Lockie Churchill.
Beautiful. Thanks so much for the introduction, Simon, and thanks, Ajay, for that really meaningful talk. And it's going to be pretty hard one to back up from that, but I'll do my best, and hopefully I keep you all interested. So good afternoon. Yeah, it's a real pleasure to be here today and see so many faces, both familiar and new, and I hope you find all these sessions, but especially mine, thought-provoking and interesting. So my name's Lachlan Churchill, as Simon just said. I'm a final year PhD student, and yeah, I want to talk to you today about predicting Parkinson's disease and finding out what's happening in these really early stages. And it's not just important for research, but it's important for how we're going to change the landscape of medications and future diagnosis. So the Parkinson's disease story, as many of you know, is incredibly complex and personal. So each patient's journey is unique. People experience a different combination of symptoms, as Ajay's just been speaking about, and they even progress in different ways. The actual pathology progresses differently from patient to patient. So there's no single typical Parkinson's disease patient. However, what we do know is that the average age of diagnosis is around 65 years old. However, at this diagnostic point, a lot of the damage has actually already been occurring, but just silently, right? So the brain's been undergoing these changes for decades before the sort of typical symptoms that onset diagnosis, such as tremor or stiffness. In fact, at this time of diagnosis, around 60% of the dopaminergic neurons in the substantia nigra are already lost. So we're still trying to uncover the exact way the Parkinson's disease starts, the combination of lifestyle, genetic, or environmental factors that cause someone to develop the disease. And so the real challenge is that at the point of diagnosis, we're in the later stages of someone's life, and we're trying to figure out how the story starts. So the big question is, in this Parkinson's disease story, can we turn back the pages? Can we uncover what happens in these early chapters before any obvious signs of Parkinson's disease develops? And this would help us understand how Parkinson's disease begins, why it develops, and how it affects the brain. And of course, that leads to, if we can understand these early changes, or even can we identify these early chapters and slow or stop the disease before it really starts to affect patients. So prediction is incredibly important in understanding Parkinson's disease. First of all, it can help us understand sort of how Parkinson's disease progresses or spreads, the sort of temporal order of the pathological accumulation of alpha-synuclein, which is the misfolded protein that causes Parkinson's disease. And also by tracking the disease in this way, we can look at when specific brain regions are vulnerable and associate them with the onset of Parkinsonian symptoms, such as visual hallucinations, freezing of gait, or cognitive fluctuations. But what other benefits are there for these warning signs? Well, if we're able to find these early signatures, and we might be able to predict the future of what an individual will go on to develop, we can then stratify patients and sort of figure out which patients are at risk for developing a motor phenotype of Parkinson's disease, or those that are more vulnerable for a cognitive risk of Parkinson's disease. And importantly, this would allow for clinical trials and the development of medications to be able to, for that specific patient, to be able to stop the onset of their symptoms. Okay, so you might be wondering about what these early warning signs are. So usually it starts with a loss of smell or constipation and potentially low blood pressure. However, these symptoms are really common in the general population. So a lot of patients who have Parkinson's disease will have these symptoms, but so will a lot of people who won't go on to develop Parkinson's disease, meaning that these aren't very specific to Parkinson's disease and are often ignored if they show up in the general population. However, we have found a reliable indicator in the form of a sleep disorder called REM sleep behavior disorder, which some of you may or may not have heard about. And the good thing about REM sleep behavior disorder is that it's highly specific for Parkinsonian disease development. So what is REM sleep behavior disorder, or RBD, as we refer to it as? So REM sleep behavior disorder is a sleep disorder where patients will act out their dreams during REM sleep. So this can range from little vocalizations to more complex movements and can sometimes result in injury to oneself or one's bed partner. So the identification of REM sleep behavior disorder as this reliable prodrome, or early stage of Parkinson's disease, as we call it, has opened the door for researchers and patients to understand more about this disease. It's kind of uncovered some of those earlier chapters that we were speaking about. So currently, 75% of patients diagnosed with isolated REM sleep behavior disorder, meaning the sleep disorder only, will go on to develop either Parkinson's disease, dementia with Lewy bodies, or multiple systems atrophy over a 12-year period. So this makes it vitally important to identify these patients and track them leading up to diagnosis. And then we can really understand what's happening at that point right before they're diagnosed. We can identify the specific signatures that differentiate someone converting to dementia with Lewy bodies versus converting to Parkinson's disease. And we can also understand the mechanisms behind what breaks those final straws right before diagnosis and leads to the onset of symptoms. So not only is REM sleep behavior disorder important in early identification of Parkinson's disease, but as Ajay was speaking about earlier, it actually forms a real important role in subtyping Parkinson's disease. What we know is that patients who have REM sleep behavior disorder and Parkinson's disease will have a more accelerated sort of cognitive phenotype of Parkinson's disease, separating those who have Parkinson's disease who don't have REM sleep behavior disorder, which is about 50% of the Parkinson's disease population. So luckily, with the help of our amazing patients and the research team that we do have tools to identify these early changes. So in our research, we mostly use neuroimaging. So we'll start with structural MRI, which Ajay described earlier. It's essentially a snapshot of the brain. We're able to look at the volume of different brain regions, which is a kind of a proxy marker for the health of cells in those brain regions. Next, we look at functional MRI, which looks at the brain's use of oxygen throughout the course of a scan. And we can kind of understand how the brain activates. And finally, we have clinical testing, so cognitive scores and questionnaires to understand the specific symptoms associated with an individual. And over the past 10 years, we've had the pleasure of having so many IRBD patients come through our clinic, and we're able to actually take these markers or these images at multiple visits. So a patient will come in for their first visit, just after their IRB diagnosis, and then maybe 6 or 7 years later, they'll come back in. Maybe some of those patients would have developed Parkinson's disease or dementia with Lewy bodies, and we can undergo these tests at their second visit. And finally, they might come in for a third visit. Maybe other Parkinsonian symptoms have developed, such as freezing of gait or visual hallucinations. And we're able to look at the differences between these markers between each visit and really understand what underlies someone being diagnosed with either Parkinson's disease or dementia with Lewy bodies, or the brain signatures associated with the onset of symptoms. So what have we found? And thanks to that, and thanks to all the patients coming through, we actually have one of the most richly phenotyped IRBD cohorts in the world. So we've actually found some really awesome research based on that has never been discovered before. So it's really, really interesting. So what have we found in our structural MRI? Well, typically, Parkinson's disease is associated with the onset. Parkinson's disease is associated with the degeneration of the dopaminergic system, which is highly responsible for the onset of motor symptoms. However, there is also a large amount of non-motor symptoms that happens in Parkinson's disease that's widely misunderstood. However, we have kind of discovered that's associated with this cholinergic system, which Anna and some of the other researchers will go in depth about later on. So we wanted to use structural MRI to check the integrity of this cholinergic system in patients who just have the sleep disorder and see if it's able to predict the onset of non-motor symptoms, such as cognitive decline or dementia with Lewy bodies. So how do we do this? We use structural MRI to look at the cholinergic basal forebrain, which is this nuclei that is responsible for innervating the cholinergic system throughout the entirety of the cortex. And what we found is that patients at visit 1 that just have the sleep disorder, no cognitive impairment, no anything, the size of their cholinergic basal forebrain was predictive of their likelihood of developing dementia and the time of dementia development. So this is before any changes in symptom profile. We were able to find a structural MRI marker that says that patient is at risk of developing dementia. And while we were discovering this, there was another group in Korea that discovered the exact same thing. So it's really awesome. And sometimes when their paper comes out before mine, I'm a little bit, but what it means is that it's real and that it's valid and that multiple people with multiple cohorts that are, made up of different ethnicities and genetic variations, they're seeing the same thing. And what that means is if we can look at the cholinergic basal forebrain and stratify patients based on their dementia risk, we can put them into clinical trials based on that. So it's really interesting. And so functional MRI, as Ajay mentioned earlier, is a little bit more complicated, but it basically looks at brain activity during a scan. And we can look at different brain regions that are active and sort of, are they active together? If they're active together, that means that they're functionally connected. And when multiple brain regions are functionally connected, they make up networks. And brain networks help us sort of undergo specific cognitive tasks or specific tasks in general. And if we can understand what this sort of normal brain organization should be, we can also identify abnormal brain organization. And the power of functional MRI over structural MRI is that in these early stages of Parkinson's disease, we know there's accumulation of alpha-synuclein. But there might not necessarily be the cell loss that we see in later disease stage. But with functional MRI, we can see the differences in how cells are behaving, not just their loss. Because before cells die, they'll start maybe firing in different ways. And we can actually capture that with functional MRI. So again, we had our patients come in, and we were looking between the visits at the differences in the functional connectivity or the brain's functional, how the brain is functioning. And what we found is that in patients who converted to dementia, there was massive differences in the brain's function in areas that control attention. And what we're also able to do, because we're interested in these neurotransmitter markers, is we're able to kind of map those changes onto areas of high concentration of neurotransmitter density. And what we found is that it was actually the cholinergic and a new neurotransmitter system called the noradrenergic system, which is responsible for arousal and sleep. It was actually the cholinergic and noradrenergic regions that were highly vulnerable in patients who converted to dementia. And that's incredibly interesting because we were able to find this before the conversion, before patients converted. We can kind of monitor these two neurotransmitter systems and, who knows, come up with treatments to stop the onset of non-motor symptoms of Parkinson's disease. And so we've got other emerging techniques as well. I'm really into the imaging, so that's kind of what I talk about all the time. But there's blood tests that detect level of alpha-synuclein. There's also skin biopsies that do the same. And finally, Jack will go through later, there's other physiological markers, such as EEG, that can look at the brain in different ways. And what we basically want to do is combine them all. And we first want to test out with big data, like RJ. We want to make sure that these signatures are valid. And once we've validated them, we want to combine them. And so the idea would be a patient comes into clinic, they get structural MRI, functional MRI, clinical tests, blood tests, skin biopsies, and an EEG. And for that individual patient, the clinician will be able to look at that patient's composite score of all of those measures and understand that individual's disease pathology, understand the symptom development in the future for that individual and treat that individual person. And also allow for the creation of clinical trials to be able to test the offset or delayment of specific symptoms developing. If we know someone will go on to develop Parkinson's disease or dementia with Lewy bodies in 3 years, for example, we can get a cohort of those patients in and test out medication so we know that it's working. And we want to change the Parkinson's disease story. So we've been able to identify some earlier chapters, but we want to change how this story ends. And that's done with no one else except you all and all the help that you provide us. So thank you so much.
Lockie. So while Jack's just setting up his slides, I will point out that Lockie is supported by research funding from the Bierzonski-Berchich Foundation. So we've been very grateful to the foundation. You'll have seen in the newsletter this year that sadly Mrs. Berchich has now passed away. But they continue to support our work ongoing, so we're entirely grateful for that. The other thing I would highlight is this cohort of people that don't yet have a disease. We're actually we're the first team in the world that's actually doing a clinical trial to see if we can change the future. So we got the money out of Parkinson's UK and working with Oxford University. We have patients who are basically getting drug to see whether we can change the future. And so, being able to back that up now and do these sorts of scans and say, well, can we see any objective evidence? Is there anything else happening here? Is going to be really vital for us. Can I just ask before Jack speaks, hands up if you think your sleep could be better? Can my team put their hands down, please? We don't need that. So Jack is basically running a massive trial, a study, where effectively we're looking at how sleep impacts cognition and what we might learn from that might be helpful for us to predict the future and better treatments. So please welcome Jack Anderson.
Thanks so much for that lovely introduction. So yeah, I'm Jack. I'm a PhD student. I'm also coming up to my last year, and thanks for joining me. I've seen some very friendly faces I've seen before and some new. And like Simon said, I want to take you on a quick tour of what happens in the brain when we sleep, how those changes are affected in Parkinson's and Lewy body diseases, and how these problems are related to thinking and memory. And then finally, we'll have a little sneak peek at some of these technologies that we're using in the lab now. But the first question I'm sure on everyone's lips here is, Jack, why are you so obsessed with sleep? What does it matter? Well, sleep isn't just rest. It's an active process where the brain does some of its most important housekeeping, as demonstrated quite clearly by our first speaker, Ajay, right there. And during sleep, the brain is still hard at work. It's essential for memory consolidation, emotional regulation, it cleans out waste products, and that's just a few of the things that go on. And it's not a simple process either. It's extremely dynamic. It's orchestrated by distinct but interconnected brain systems and pathways, some of which are heavily affected in Lewy body pathology. So sleep, it's not optional downtime. It's essential maintenance for the brain. And sleep problems, they're very, very common in Lewy body diseases, like you've all demonstrated. One review I was reading recently found that up to 98% of people with either Parkinson's disease or dementia with Lewy bodies experience some kind of sleep disturbance. So a well-known example is REM sleep behavior disorder that Lachlan's just given us a great overview on. It's actually one of the core features in diagnosing DLB. It's seen in around 80% of people with DLB, 50% with Parkinson's, and this is a sleep disorder. And it's not the only one. We have insomnia here, excessive daytime sleepiness, restless legs syndrome, and that's just to name a few. And there's a real problem here because these sleep problems are described just based on symptoms. They're described just their clinical features, the underlying brain mechanisms that are involved here. It's not really well understood. And this is an important gap because these sleep changes appear sometimes years before any movement or memory symptoms. And for me, this really strengthens my advocacy for sleep research. It's an incredible window into the brain. You know, when we're awake, our minds are constantly bombarded with external stimuli. What am I going to have for dinner? That driver's going really fast. What am I having for dinner? But when we're asleep, the brain is free from those distractions. Some say that sleep is when the brain's at its most honest. So what's actually happening when we're sleeping? Well, as many of you know, every night our brain cycles through different stages of sleep. It begins with light sleep and moves progressively into deeper stages, eventually reaching slow wave sleep, the deepest stage where the brain activity is the most synchronized. And after that comes rapid eye movement sleep, or REM sleep as we call it, where brain activity speeds up and it almost looks like wakefulness, except our muscles are relaxed and we don't act out our dreams. But that's an obvious problem here in Lewy body diseases. So these cycles repeat roughly every 90 minutes or so, but they can become very fragmented. And I've always been fascinated by neurophysiology, or at least that's what I tell people in the pub to try and look cool. But you can see here that as we move through sleep stages, this is how brainwaves are kind of seen on EEG. So it starts very fast frequency, short amplitude, and as we move into deeper stages, you see these big, slow waves come up. And finally, when we get to REM sleep, it becomes really, really, really fast again. It almost resembles wakefulness. And these waves can tell us a story about brain integrity and health, and has been a real focus of my PhD. And we all know that sleep and thinking are really tightly linked. After a bad night, our memory, focus, our mood, all of these can be affected. And in Parkinson's disease and Lewy body diseases, these effects can be magnified and may be caused by the underlying changes that are happening in the brain. It's almost a bidirectional relationship here. And of course, like sleep problems are multifaceted, but if we just think about REM sleep for a second, the brain circuits that shape REM sleep are also the ones that support cognition. And that's the cholinergic system, which we touched on earlier, and Anna, my colleague, is going to go into in more depth a little bit later. And we know that this system can be affected in Lewy body diseases. So this question really interested us and made us dig a little deeper and try and confirm this link that's not been shown before. And as I said, there's not a lot of research done in objective sleep, definitely not in Parkinson's disease and Lewy body diseases. So in our research, currently, we're exploring how changes in sleep, particularly in REM sleep, that dreaming stage, reflect what's happening in the brain. And what we found is that people with thinking and memory problems, they tend to show changes in brain activity, a slowing of that REM sleep signal that I showed before. And what we found is that change in signal is correlated to memory and to cholinergic brain systems using MRI, using functional MRI, like Lachlan was talking about. So what this tells us is the way the brain is behaving during sleep gives us a window into what's happening when we're awake. REM sleep, as an example, could help us one day detect or track changes in the cognition earlier and more easily, and potentially could help us guide and see how treatments are affecting. And that was just some beautiful graphs and plots. That was just to show off. You don't have to worry about them. But I think the question, again, that's on everyone's lips is, how do you measure these brainwaves? Like, I'm quite good at this. I ask a question that I think you're going to answer and do it myself. It makes my life really, really easy. But what we use is high-density EEG. So it's lots of sensors that are placed around the scalp to record brain activity in fine detail. We do this in wake, and we also do it in sleep. And here's an example of a real participant, 2025, one of the most rock and roll men I've ever met, that was in our study. Because it captures signals from so many places of the brain at once, it's not just a change we can see, it's where and how. So in Lewy body diseases, these changes can have a spatial difference. So being able to understand that is really, really important. So like Simon mentioned, we're currently running a study at the Wilcock Institute, which is using high-density EEG alongside MRI testing. We've got some representatives from our imaging center at Macquarie. And we also use overnight sleep studies and a really detailed cognitive battery. Patients, they stay for 2 nights. And on the second night, we do a partial sleep withdrawal, where people's sleep is deliberately disturbed. So we want to see how the brain adapts to sleep loss and whether it struggles to adapt in Lewy body diseases. It is the most detailed study of its kind. And the main researchers involved on the ground are myself, Lachlan, and Ajay. So it's really just a 2-day comedy show with a little bit of science we put in between. And we've been doing this in IRBD patients. I won't point him out, but there's a gentleman in the crowd that was sleep deprived this morning. So both him and I are a little bit tired. But we've also done this in Parkinson's disease and dementia with Lewy bodies. So anyone can really get involved here. And it looks a little bit like this. It's normally me and Lachlan with 5 or 6 screens and a high-density EEG. It's a lot of fun. And our goal is to use sleep as a window into brain health because, like I said, these sleep changes can happen years before any other symptoms manifest. So EEG might be able to help us identify people at risk or those likely to benefit from early intervention, especially when it comes to cognition. And if anyone's interested, feel free to come find me in the break. We can have a little gossip. But I think something that would keep everyone engaged is we do a real quick quiz, just to make sure people weren't listening. I wasn't boring anyone. So firstly, in my research, slowing of brainwave signals and which stage of sleep was related to cognition in the cholinergic system. Feel free to shout out. Which stage was it? REM sleep? Yes. Everyone's right. Beautiful. And probably most importantly, which researcher from our team looks the best in high density? Shout it out. It's a joke, but no one's saying an answer. Like it's quite obvious. It's me. It's me. And if anyone have their phone out, feel free to give this a scan, because this has all the information to our study, and people can get involved if they'd like. My email's there as well if anyone's had any questions. But thanks so much for listening, and we couldn't do any of this research without you. Yeah, and that's me.
Wonderful. So whilst I think Chris is setting up his slides, oh, sorry. I was going to photobomb.
His favourite.
There you go. Yeah, no favourites, no favourites. Many people in the room will know I was pretty late to fatherhood, and I'm now understanding what it means to be proud of your children. But this part of my life has been really special. Some of you might notice a gender equality. I promise you in the second session we will balance the gender inequality. It's important for me and important for you too. Our final speaker for this session is Chris Coppin. So Chris is a qualified neurologist who is doing his PhD with me. I've asked him, however, not to talk about the stuff he's doing in his PhD so much as some of the other work that he's helped put together, which I think is going to be very important clinically. And we have published a bunch of this data, but this is, I think, one of the presentations I think is really, you're going to go home tonight and think about and say, is that me? Please welcome Chris Coppin.
Thank you. Thank you, Simon. I think you're like running the prices right with that microphone. You know, come on down. So my name's Chris, as he said. Out of the research and into the clinic a little bit with this talk. So today I'm here to talk to you about bones. Very exciting topic. We all have bones. We've all used our bones over time. And bones are very important for not falling over. If you don't have bones, you would be sort of a puddle of flesh on the ground. And so they are really important for us. But they're also really important not to fall over, to take care of your bones. So why is that? It's because of osteoporosis. So I'm sure you've all heard of this before, but we wanted to take some time to talk about what is osteoporosis, what does it really mean, and how are we looking at this in the clinic. So osteoporosis is a condition where the bones become thin and weak over time to the point where if you were to fall over, you'd be at a much higher risk of breaking something. Medically, we define this as a bone mineral density of less than 2.5 T-score, which for any maths enthusiast people is 2.5 standard deviations below the mean of a healthy young person. So if you are a bit older, you're already at a disadvantage, but we're really looking for those people who are really quite low. Where the bone density is decreased, but not quite as bad as osteoporosis, you may have heard that called osteopenia. It is actually reasonably common. 43% of women over 70 years of age will have osteoporosis. Only 13% of men. Unfortunately, this is a hormonally related condition. This is what it looks like on a laboratory slide. You can see normal bone there on the left, thick cortex coating on the outside, and then a honeycomb trabecula in the middle that makes the bone strong but relatively light. On the right, you can see there is thinning of that cortex at the top and bottom with holes and almost loss of that trabecular honeycomb reinforcement through the middle at times. Here in a 3D scan view, you can again on the left see a normal, strong honeycomb structure, but on the right there in someone with osteoporosis, you can see a lot of that structure is lost, and what is still there looks like it could break quite easily, and indeed it could. Why does this happen? It's because bones are living organs. They're not something that are built when you're growing and becoming taller as a child and then freeze. They're in fact turned over and indeed completely replaced almost during life. The way this works is through a cycle of cellular activity that is demonstrated pictorially here in this picture. What happens is that these blue osteoclasts here towards the left of that picture come along and eat up some of that old bone. They are then followed closely behind by the purple osteoblasts, which come through and lay down new bone. This ensures the bone is always fresh and strong. It also is how your body, one of the main ways your body controls calcium in your blood, by releasing calcium by reabsorbing bone. Calcium itself then being important for things like nerves and brains and slightly more interesting organs like those. All of this process does require an adequate oral intake of calcium and vitamin D, and that's where you hear things like making sure you get enough calcium in your diet and enough sunlight to produce vitamin D. This metabolism cycle, however, changes with age. The natural progress of your bone density over life is that it grows as you do. through the early years of life before peaking in the 20s and 30s. It then tends to decrease over time as those osteoclasts who are taking the bone become a bit more active than those osteoblasts that are putting down new bone. Unfortunately for women, this is worse straight from the start as they have a lower peak bone density, and then it accelerates even faster for them after menopause. And this leads us to a thought that some people are more risk of having osteoporosis, of that bone density really going down, than other people. What puts you at risk? Well, it's age, for one thing. As you saw with those curves, it decreases with time. The other thing, as I said, is gender. Estrogen is protective for bones, and estrogen post-menopause drops faster in women than actually in men. Medications can cause problems, such as long-term steroids, and also chronic health conditions, such as kidney health disease. Alcohol and smoking can also make this worse. Your level of physical activity is important, both before that peak onset, because if you're as a younger person doing weight-bearing exercise will achieve a higher peak than those who don't. But then also through life, if you continue to do impact exercises, you will maintain a higher level of bone density than if you didn't. And then we put on top of that, if you already have osteoporosis, the risk is if you fall over, and we think about conditions that make it more likely for you to fall over, which would be things like Parkinson's disease. So it next comes to fragile. So this is a project that we've undertaken with some research students who have visited us from the University College in London. I don't think that's Doc's preferred University College in London, not Oxford, but they were very helpful with this project. You may have been asked about osteoporosis in the clinic, and this is part of what we were doing. So with this project, we tried to identify factors in the clinic that would make it more risk of someone having an osteoporotic fracture. So both having osteoporosis, but also going to the point of having a fracture. So in the process that we've been talking about, the important things would be things like risky medications, those long-term steroids, institutionalization and the effects that can have on diet and exposure to sunlight, amongst other things, and also other long-term conditions like diabetes and bowel disease, which can affect the calcium metabolism. But we were also thinking about activity to promote bone health and also gait problems and things that would make it more likely for you to fall over if you did have osteoporosis and have a fracture. As part of this, we wanted to look at how many patients we were sending off to have their bone health checked. As part of a weirdness of Medicare that we're all used to, if you want to have a bone health check, it's through the GP. So we were looking to see how many people we sent back to their GP to have this looked at. Last year, in sort of the early last year in late 23, we did okay. We referred about 23% of the people we should be referring. This year in the same time period, we referred 48%, so over doubling, which is good, but there's still a way to go. But what can you do about it yourself? So the important thing here is to find out. If you don't know how you have osteoporosis, you can't do anything about it. Osteoporosis can be confirmed with a simple bone mineral density scan. This is sort of just a fancy X-ray that can be ordered through the GP, and it's generally done over 2 years. So if you've had one in the past but you can't really remember when it was, it might be time to have another one. We also assume someone has osteoporosis if they have a major fracture, which generally means something like a hip or a spinal vertebra. If osteoporosis is confirmed, we generally look at drug therapy. And there's a number of ways we do this. Coming back to the mechanism, these mechanisms that recycle your bone are the same mechanisms we target with these drugs. Common ones you might have heard of Prolia and things like bisphosphonates, alendronate, risedronate. These drugs act on the osteoclast to stop them eating quite so much bone, which allows the osteoblasts to catch up and build more bone, and over time push things back in the stronger bone direction. There are other drugs that push bone growth directly, like rosumab and teriparatide, but these are more of a specialist endocrine clinic thing. The other thing that can be done as well is to replace some of that estrogen in postmenopausal women to improve bone health. I will point out what's not on this slide is calcium and vitamin D. Though they're important for bones, they are not directly treatments for osteoporosis. So with osteoporosis, we'll tell you to get enough calcium, enough vitamin D, but that's not a direct treatment. In summary, number one, don't fall over. We try and do our best to make your bones as strong as they are, but if you're going to fall over, it's going to be a risk anyway. So those things like falls prevention strategies, those core strength and balance, those things are still very important. Keep exercising. So again, core strength and balance helps your balance in Parkinson's disease, keeps you upright. But also for bones specifically, weight-bearing and impact exercises are important. These are a bit harder if you've got arthritis and health conditions, of course, but these are things like running and sports and things where you're actually hitting the ground with some force. It is still important to supplement the vitamin D and the calcium so the bones have enough materials to work with, but again, it's not a direct treatment. And of course, talk to your GP. They can talk you through these options and make sure you get the bone mineral density scan if you haven't had one. And that's it. Thank you.
That's just wonderful. Can I ask our other three presenters to join me and come on this right-hand side? We have two roving mics on either side of the room, the guys who've got the microphone. So if you have questions that you want to put to the panel, the motley crew that they are, then please do. And at home, thank you for some of the questions that have been coming through. Please keep them coming through. Ajay, first one for you. I think hopefully that will reach. Yeah. People want to know, how far off are we from going in, having a brain scan with your doctor and saying, I am group A, B, C, or D. I'm the person who's going to get no dementia. I'm the person who's going to get dementia. Are we at that point yet? And if not, how do we get there?
Thank you so much for asking that question, because this is something I can answer. And
I didn't ask it. It's come through.
So I wouldn't say we are very close, but we definitely made a huge step when we found out that there are 4 subtypes within Lewy body diseases. But again, as I mentioned in my last slide, that there are a lot of things that we still don't know about. Like we don't know how the brain talks to each other. We don't know about how, in each of these subtypes, we don't know how the brain connections are within these subtypes. So we need to get a better idea of, a complete picture rather, of the disease to kind of like say, hey, a patient is visiting a clinic and then he just gets a complete battery of the testings, which they usually do for other diseases, but we just don't have it for Parkinson's disease. And if we have all of the results, then we can just run a simple technique and then say, okay, this patient is subtype A, and he is at stage 10 right now, but by the time he reaches stage 25, he might develop cognitive impairment. So which is essentially important to look at his brain regions that usually are implicated with memory and stuff. So,
and I think just to add on to that, it's been a big initiative all across the world to have these really large-scale multi-center sites that are being started up in the US and whatnot, where they do collect brain images, they do collect pathology, they do collect skin biopsy, they do collect blood tests. So I think what it will be is, right now, we're at the point where we're looking at these big data in their individual, structural MRI or functional MRI. But I think once that's all validated and we have these massive cohorts where we can look at it across the modalities, that's when we're really going to be able to have someone come in and be able to understand what subtype or group that patient's in. Yeah.
And you've got the microphone, but if there are any questions, please raise your hand and we'll get a microphone to you. So we've got a question's going to come here, Nicole. While we're getting the microphone there, just wait for the microphone because we're doing the live stream. Before we answer that, again, the question is, if I go to my doctor, can I get my cholinergic area scanned and be told whether I'm going to be someone who gets dementia or not dementia? Are we at that point?
So what we need is, right now it's being validated across international centers, but what we actually need is we need a large scale of the population to actually get their cholinergic basal forebrain imaged so that we know what is normal and what's abnormal. Because right now, in this study, for example, we did it at a group level within all the patients in our cohort. But for a patient to come in and have their brain region individually scanned, we need to know what the general population is normal, so then we can truly identify people that are at risk.
It's a bit like when we've got blood sugars and you had to work out what was normal.
Yeah.
Sir.
Very quick question. What time scale are you talking about?
Yeah.
in years, as an example, to be able to have this data to then to be able to, say, the two of you put that together and review it?
So, for example, in my study, when the patients were coming in, they came in over about a 5 or 6 year period. So we'd have a patient come in, and then maybe 4 or 5 years later, we were able, like from that point, we were able to predict their prognosis at a 4 or 5 year period.
Can I add that? So in addition to that one, so when you say time period, so I didn't put it up in my presentation here, but it would be part of my papers. So we followed up clinical data for 15 years. So these were patients with Parkinson's disease. And obviously, we all know that predicting dementia in Parkinson's disease would be a huge development, and which people have been trying to address, but we couldn't. So we had data for when we followed up patients for 15 years, and we were able to identify one particular subtype that's extremely at higher risk of developing dementia. So, but again, we just did it on clinical data sets, but we have to validate on other data sets to kind of like give you an estimated, kind of an update of where we are at this point.
Ruben, can I just ask you to keep rolling the slides just in case people missed the QRs from earlier? That's great. It's the obvious, easy question. How do I improve my sleep?
It's a really, really, really good question, and it's something that we.
I wasn't them. It's for me.
Yeah. Work less. But yeah, there's lots of obviously lifestyle factors. The hard things when we talk about the conditions, diseases we're interested in, is there is that bidirectional relationship I was talking about areas of the brain that are involved in sleep and Parkinson's disease. There's an overlap there. Obviously, there's been large-scale clinical trials on different medications. I speak to a lot of patients with IRBD that use things like melatonin. The large data says that there was no significant differences, but at a patient level, I've seen people say improvements. But the real thing I would say is a lot of lifestyle factors, like say keeping up with exercise in the day is a really big one. Trying to keep your circadian rhythm as best you can, so going to bed at reasonable bedtimes. But it's a really, really difficult question.
I think you did well. We've got a question just here. James, do you want to just see? Oh, Nicole, are you there? Yeah. There you go. Perfect.
Regarding sleep, was there some work done a few years ago about whether CPAP improved the quality of sleep?
So the question is about whether treating obstructive sleep apnea with the CPAP machines, blowing things in. I'll take this as a clinician if you want to take it as your...
Well, I just wanted to make a point quickly, is my brain kind of did a little backwards there. Was that a friendly accent I heard?
It is a friendly,
yeah,
it's
well,
a friendly accent.
Yeah, like my brain kind of just did this little like...
The question came with subtitles.
Yeah, I think I went back in time.
Your understanding is that it's friendly. I think you've been away from home for too long.
It's wonderful.
But with him, I'll get you to comment, of course, Doc.
Please,
but with CPAP therapy, we do have a lot of patients using CPAP therapy. It's really good for consolidating fragmented sleep, especially for people that have obstructive sleep apnea. Their AHI is relatively high, which is actually more common in Parkinson's disease than the general population of their age, just due to weakening of the throat and other factors as well with immobility. So yes, it definitely is something that I'm sure you recommend. Being a neurologist working in a sleep clinic.
Yeah, so the studies out there are actually a bit weak. The one study was kind of correlational, looking at sleep apnea and Parkinson's disease, and it suggested it was helpful, but it didn't. It was more on cognitive, so it helped the cognition, which is not perhaps surprising. The biggest studies looking at sleep apnea in non-Parkinson patients, but patients who are at risk of dementia, weren't that strong in terms of reducing the risk of dementia. But for all the reasons that Jack points out, if you've got sleep apnea, I mean, the trouble is our disease, Parkinson's, makes people sleepy. Our drugs make people sleepy. And if you've got sleep apnea on top of that, you're going to be sleepy. You don't need to be that. And of course, then you get the other seven dwarfs kicking in. There's grumpy and there's very little happy. Chris, someone's asked us about the calcium, vitamin D, but is there anything else that we can do to healthily eat our bones stronger, as it were? Because exercise isn't popular, it seems, it turns out. Is there a pill for exercise?
The exercise is difficult for osteoporosis because, again, it's that impact exercise. It's playing netball and running on hard surfaces and things that just make you feel uncomfortable just thinking about it. But and it's also just harder with the conditions that we deal with. From a diet point of view, Not hugely. It's really making sure you have enough calcium. If you don't, then looking at calcium supplements like your Ostalin and those sort of things you've probably heard about. Vitamin D is interesting because it's part of your diet, but it's probably not something you need to target. But then it's about getting enough sunlight to activate that vitamin D, which doesn't take that long. It's sort of 20 minutes out in the sunlight, not really at the peak of day. But otherwise, take it in supplementation, particularly for people, say, in nursing homes, not getting outside as much as they could otherwise.
Are there any other questions from the floor? I think we've gone through everything that's coming online. There's one just here. Thank you.
Just to the first couple of speakers, a lot of the research that you're doing is about predictive diagnosis. Are there benefits for people who already have Parkinson's in your research and in the data that you're collecting?
So I think, speaking on my research in the REM sleep behavior disorder, I think this arm of research is really about predicting it early for those patients who are yet to develop Parkinson's disease. But also, if we're able to predict symptom development, then we may still be able to help people who have Parkinson's disease but might not have developed, for example, freezing of gait or visual hallucinations. So we can change it from a bit of a diagnosis target to a bit of symptom development target. So I think that's...
Yeah, just to add that part, in my study, we found that cognitive impairment and visual hallucinations are particularly associated with one particular subtype. And so people might present with cognitive impairment early on, but they don't develop visual hallucinations until a later stage. So we might be predicting visual hallucinations for that people, and it is a huge burden. Like visual hallucinations is a big burden. So if we are able to like predict visual hallucinations just by the MRI scan at baseline, it would be a huge win for us.
And I think with the clinical trials aspect of things, there might be a specific drug that targets the freezing of gait pathways. And so if we're able to subtype people or predict someone developing, for example, freezing of gait, we can maybe help those patients through clinical trials of that symptom.
Jack, did you want to add anything?
Well, it was a very similar point. It was the idea that specific symptoms, even with Parkinson's disease being diagnosed, they do tell stories for the future. Things like REM sleep behavior disorder, we speak about it isolated, but it's also found in Parkinson's and can develop after Parkinsonism has started. So being able to stratify people to give more targeted personal treatment, I think, is another arm of the research that we do.
There's quickly something that's come in, which is what's the difference between delusions and hallucinations? Hallucinations is seeing things that aren't real, or other sensory modalities, smelling things that aren't real. Delusions are believing things that aren't real. So, and it's a very limited repertoire. Someone's stealing my stuff, someone's poisoning my wife's having an affair, why are you pretending to be my wife? So it's very, nobody ever wants to be Jesus Christ or, you know, a rock star. We're going to take one quick question and then hopefully a quick answer. Hopefully we've got the microphone on. Microphone apparently is on. You just have to speak. I can't.
Yeah, I was just wondering if you have an upper age limit for your research.
So the quick answer.
Do you get to an age where you say, well, you're no good to us?
So the answer is no. And remember, we also run the brain donor program. So it's fair to say we do the isolated REM sleep behavior disorder. We do the cradle to the grave. Basically, we do all of it. And it's quite funny because some people ask me, don't you get bored? You know, you're a neurologist, you just do one disease. And I kind of go, well, come and have a look at this. Trust me, no time to get bored. So our research, I was very age activist. So the clinical trials, Australian Parkinson's Mission trials, go up to a maximum of 80. They wanted it 70 when we started. And I said, no, no. And then they wanted 70. No, 80. So we got 80, which is really high for any clinical trial. In terms of our research, the bottom line is that most people with isolated REM sleep behavior disorder are presenting in their 50s, 60s, maybe 70s. The people with Parkinson's dementia are often going to be in their 80s. So it's fair to say we are a very broad church, and the bottom line is that, as I hope you've heard from the guys today, we can't use guinea pigs. They don't have our disease. The only creature on the planet that has our disease is Parkinson's, is humans. So our guinea pigs look a lot like you. And if you're sitting there going, well, I don't have Parkinson's, I'm a spouse, I'm a carer, or whatever, we need the controls too, because you have to be able to compare. So nobody is safe in this room. Even the guys from MMI who do all our imaging, it's great to have you here. So what we're going to do is going to break. Hopefully there are some refreshments outside. I didn't check, but hopefully, Louisa, there are some refreshments outside. Apparently there are refreshments outside. Have you got the microphone? Yes, sorry.
One quick question. HRT, does that affect any of this?
So HRT obviously is not a Parkinson-specific treatment. What's interesting, and we don't want to go too off-road, is more men than women get Parkinson's disease. And the question is whether girl hormones are protective or not against the disease, or whether there's something about the mitochondria, the power cells inside cells, which come from a female lineage. When we looked at doing HRT as a therapy in clinical trials, we realized there was going to be a problem. It's very hard to blind men getting boobs. So in actual fact, it's very difficult to study in that way. Obviously, in terms of osteoporosis risk, coming back, Chris, do you want to comment on osteoporosis?
Yeah, so again, HRT can be used as an adjunct or a side treatment for osteoporosis in women postmenopausal where they're losing estrogen, and there is some thought that estrogen can push hormone replacement therapy. So generally replacing estrogen would be the main type, and it can be used in some cases.
Lovely. Okay, so look, I think we probably should break for those refreshments. I'm told that they are there, which is great. I think we give ourselves 50 minutes or so. That should be plenty of time. You know where the bathrooms are. If you're watching online, that means we're going to get back here at about, is that 10 past 3? Yeah. About 10 past 3. Please tackle these guys to the ground. Can you give a warm round of applause to them? And we'll see you after the break.